This is a lecture on protein structure for Dr. Watts' class at UAB. To get a general idea of proteins, there is a lot of information that we need to cover first. Proteins are one of the macromolecules. They are composed of amino acids. They are connected by peptide bonds. These bonds have no movement and no flexibility within them. You can have 160,000 combinations with 4 amino acid proteins. The average peptide protein is about 400 amino acids. There are less than 20,000 proteins in any given cell. The function of proteins is for flexibility and stability. Proteins will fold spontaneously as it's being built. This is a general picture of a protein shown up on the right. As proteins synthesize, they must fold to be able to fit in the space that they are in. As they start to fold, they fold into a globular formation or a fibrous formation. This is shown as linear. In folding, amino acids that are hydrophobic will move to the interior of the protein. The amino acids that are hydrophilic will arrange themselves on the outside of the protein. There is directionality to protein structure, which will be shown on the next slide. Directionality helps to organize and understand amino acid structure. With directionality, it will help orient ourselves into viewing a protein. Every protein will have an N-terminus and a C-terminus. The N-terminus is where you have an open amino group. This is not bound to any other carbon. The C-terminus is the carbonyl terminus. In this image on the right, you can see up on the left of it the N-terminus is free. On the lower right of the image, you have the C-terminus, which is represented by the carbonyl. You will always read proteins N to C. This is important. There are 4 basic structures to protein. This is showing primary structure. Primary structure focuses on peptide bonds which connect amino acids. They refer to amino acids in a linear way where you can identify the N and C terminus. R groups have no play in primary structure. The image on the right shows a basic primary structure of a protein. You're able to see the individual amino acids. Secondary structure is also a basic structure of proteins. This occurs when you begin to fold primary structure proteins. Secondary structure engages weak hydrogen bonds to help stabilize it. R groups are not engaged in secondary structure. There are 2 common structures showing early folding patterns. There is a beta sheet, which is shown on the right of the image, and an alpha helix on the left. We will now go into beta sheets, which are a part of secondary structure. On the left of this image, you can see a wavy line of amino acids all bonded together. When we go to the right of the image, you can still see the amino acids in a wavy line. There are vertical bonds in this image. These show the hydrogen bonds between carbonyl, oxygen, and hydrogens from the N-terminus. This is hydrophobic. The alpha helix is also a part of secondary structure. On the left of this image drawn, you can see amino acids in a coil wrapping around themselves. To the right, we can still see these amino acids in curls. We can see vertical bonds as well, where every 4th amino acid is bound to the first amino acid. This is done by weak bonds and shows a cross-linkage. We also have these offshoots on this image. These are R groups pointing outwards. Again, R groups do not have any function in secondary structure, but we are able to see them. By now, we can see that different structures will build upon themselves. For tertiary structure, this is the most variable in all of the structures. Tertiary structure is a completely folded protein with 1 N-terminus and 1 C-terminus. Primary structure will always be present within tertiary structure. Here we finally see that R groups have a function. In tertiary structure, R groups will cross-link to stabilize through weak bond interactions, hydrophobic interactions, hydrogen bonding, and ionic bonding. Disulfide bonds are created by covalent bonding between R groups of 2 cysteine amino acids. On the right of this image, we are able to see a tertiary structure. We can see beta sheets and alpha helixes. Quaternary structure is the final structure we will talk about in this lecture. This occurs when you have 2 or more peptides that interact together. Weak bond and hydrophobic interactions cause this. To review, a monomer is a single peptide, 2 monomers is a dimer, 3 monomers are a trimer, and many monomers is a polymer. We can see a quaternary structure here on the right. We will go into the basic domains of this in the next slide. Domains can easily be seen through quaternary structure. Domains tell us more about the overall structure of the protein. Domains will form to make proteins larger. The functional component of tertiary structure is also domains, and this is less than 150 amino acids per domain. Domains look like offshoots and have hinge points. These allow the protein to bend, allowing for flexibility. Different domains can also have different functions. If a protein needs additional support, it will add domains through peptide linkages. In this image, you can see that each color will represent a domain, and they are all linked. To do a recap of this lecture, we will do a quick review. Primary structure is a sequence of amino acids by peptide bonds. Here you are able to identify the N and C terminus. Remember, you always read N to C. For secondary structure, this deals with hydrogen bonding, alpha helix, and beta pleated sheets. For an example, keratin is composed of a lot of alpha helixes. Tertiary structure is where we start to see R group interactions having a function. Tertiary structure deals with R group interactions, disulfide bonds, hydrogen, and ionic bonding. Quaternary structure deals with domains. R groups also have a function here. In the image on the right, we are able to see an easy breakdown of the different structures and how they differ. To review domains, it tells us more about the overall structure of proteins. Domains form to make proteins bigger, and they are less than 150 amino acids each. These are offshoots, they have hinge points, and each domain can have a different function. Domains will be added onto each other by peptide linkages to make the protein larger. Remember, proteins go through self-assembly. There's no energetic cost to building proteins and building upon themselves. Proteins have the ability to function by bonding to other molecules. There are very specific binding sites for these, with high specificity and weak bond interactions. At the intersections of domains, you will find binding sites.